So one of the interesting challenges of genomics is that we're no longer looking at one gene that we're particularly interested in a study. We're looking at the entire genome, and the problem and the opportunity in studying the entire genome is a lot of health risks and benefits are buried in that. And so although you may be studying, let's say, heart disease by looking at the entire genome, de facto you're also studying all the psychiatric genetic factors. And you might find that you have an individual in your heart study cohort who actually has a strong risk for major depression because of some mutation. And these incidental findings may actually either be truly false positives or irrelevant and should be ignored, or these incidental findings may actually be very, very relevant to the patient's health. And this is increasingly something we're going to have to grapple with in this era of genomics where we're measuring a large fraction of the genome. As you probably know, there are companies out there like 23andMe and Navigenics, which are now doing whole genome-wide scans. And very much like these MRIs that are offered as birthday presents, in my mind, incautiously, when you look at everything, as in when you do a total body MRI, you might find something that you did not want to know or you will not be able to interpret. And so I think the responsible view of how to interpret these results is in the context of genetic scientists who are able to look at these large databases, really ferret out whether these mutations are in fact meaningful, whether they are likely to cause disease or offer benefit, and then submit to the IRB the opportunity to communicate to these patients.